I guess I want to start off by thanking everyone for coming and attending my exit seminar. I've spent a lot of time working on and thinking about this project, and I was lucky enough to be able to conduct all the field work here last summer. And so it's really nice to be able to host my exit seminar here. It feels like kind of a full circle event. I'll start off by introducing myself. As Steve mentioned, I completed my undergrad here as well, doing a thesis in Adam Chippendale's lab looking at fruit flies. And I made the switch to frogs in September of 2020. And like many of you, I'm sure I've always loved spending time outdoors, and I've always been particularly interested in frogs. So I've been catching frogs since I was in diapers, and I used to really love setting up buckets, or in this case, my turtle wheelbarrow, with some water and rocks and adding some leaf litter in, and I would call these my habitats for the frogs. But what I realize now is I was actually setting up mesocosms. So I have been kind of prepping for this study for my entire life. And as Steve mentioned, what really attracted me to this project was the ability to kind of use science to contribute, even in a very small way, to the protection of the environment and the protection of frogs. My research is motivated by the problem of oil sands process affected water, or OSPW, in the oil sands region of Alberta. And OSPW is produced as a byproduct of bitumen extraction and held in these gigantic reservoirs referred to as tailings ponds. So OSPW can enter the environment through seepage out of tailings ponds and into groundwater, and then subsequently other bodies of water, as well as through accidental spills that happen after pond breaches. And for these reasons, new policies are being developed that will allow for OSPW. Sorry about that. There we go. Okay. Sorry, new policies are being developed that will allow for OSPW to be treated and released into the nearby Athabasca River. And despite these plans coming into effect in the next couple of years, the ready to release criteria for OSPW has yet to be defined. And these introductions into the environment are of concern because OSPW contains high concentrations of a number of different contaminants, including salts, metals, and organic compounds. So I think in order to conceptualize this problem, it's helpful to look at a map. So this shows the Athabasca region of Alberta, where a lot of oil mining and subsequently a lot of these tailings ponds are located. So here, running through the middle, is the Athabasca River, which flows from the south to the north. And there are currently over 500 billion liters of OSPW being held in these tailings ponds. And if you look kind of down here at the scale, you can see that many of these are within a couple of kilometers to the Athabasca River and are also located just north of Fort McMurray, which relies on the river for drinking water, and are also upstream from a number of provincial parks and important wildlife habitat. And this is that same image, just blown up slightly. So these tailings ponds are also located within close proximity to a number of Indigenous communities, which may be disproportionately affected by oil sands pollution. And each pond here, indicated with a red color, has been given an extreme consequence assessment, meaning that a substantial spill of water from this pond would result in a substantial loss of human life and irreversible impacts to wildlife habitat. So obviously, this is kind of a problem that needs solving. And as I mentioned, OSPW contains high concentrations of a number of contaminants, one of which being naphthenic acid fraction compounds, or NAFCs. So naphthenic acids are carboxylic acids that occur naturally in crude oil, And NAFCs is a term used to describe the complex group of naphthenic acids that are found in OSPW. And they're solubilized into this wastewater during that oil extraction process. And NAFCs are important because they're responsible for most of OSPW's toxicity. And as such, understanding what levels of these compounds are toxic is important to informing OSPW release plans, as well as kind of environmental monitoring plans. However, the safe water or water quality guidelines for NAFCs have yet to be developed. And with my research, I study NAFC toxicity in frogs in order to help inform the development of these guidelines. So frogs are important bioindicator species for the health of aquatic ecosystems. In many species, reproduction and early life development occurs in small ponds and vernal pools, which can accumulate high concentrations of contaminants. And both events are under tight hormonal control, and so frogs can be helpful to study the endocrine disrupting abilities of many chemicals, so chemicals that disrupt the hormone system. Also, unfortunately, amphibians are one of the most at-risk groups of animals on the planet, and so studying them and understanding how they respond to chemical exposure is important to kind of understand what risks things are putting these populations at risk. Wood frogs specifically, I'm just not going to touch that. Wood frogs specifically, my study species, have a very large North American range, and they're one of the most common amphibians in the oil sands region of Alberta. They also are explosive breeders and breed reliably every spring, and their offspring develop from embryo to metamorph within 6 to 12 weeks, meaning that multiple life stages can be assessed in a single experiment. So for this reason, frogs have been used to study OSPW and NAFCs in the past. Wood frog tadpoles raised in reclamation wetlands formed from OSPW suffer reduced growth, increased mortality, and are less likely to successfully reach metamorphosis. And these individuals also show abnormal thyroid hormone ratios, suggesting that OSPW may have some endocrine disrupting effects. Western clawed frog embryos exposed to NAFCs in the lab show similar responses, as well as an increase in developmental abnormalities, including those pictured here. And adult Western clawed frogs exposed to commercial naphthenic acids, which are similar but not exactly the same as NAFCs, show reduced calling behavior as well as mating success, as well as reductions in androgen-related gene products, again suggesting that NAFCs may be disrupting the endocrine system, in this case through anti-androgenic activity. However, these studies have mainly been limited to brief early life exposures to these commercial naphthenic acids, which are not entirely representative of NAFCs that are found in OSPW. And to our knowledge, no studies have assessed the reproductive behavior of these NAFCs in frogs. So the aim of our study was to kind of address that gap and assess the potential for NAFCs to affect reproduction in the wood frog. I'm just going to take a sip of water. So in order to address this aim, it's important to look at the mating event as a whole. So wood frogs arrive to vernal pools in early spring. They mate and enter amplexus, which is the mating behavior shown here, which is basically just a male clasped dorsally onto a female. Females then oviposit eggs, and males fertilize them externally. These eggs hatch, and tadpoles develop, eventually emerging from the pond in mid to late summer through metamorphosis. So therefore, a successful mating event includes one, both where frogs are able to mate and are fertile, as well as able to produce viable offspring that can emerge from the pond. So considering this, my project had two main objectives. The first, to determine the effect of exposure on reproductive behavior and fertility of adult wood frogs. And the second, to determine the effect of exposure on offspring viability. And then I had two corresponding hypotheses. The first, that NAFCs would interfere with reproduction through behavioral effects and decreased fertility. And the second, that NAFCs may interfere through reduced offspring viability. And I addressed or tested these hypotheses in a 4-month mesocosm study conducted at our mesocosm facility, which is just up the road on Warner Lake. And this experiment consisted of a mating behavior and fertility phase and an offspring viability phase. So during the first phase of the experiment, we exposed wild adult wood frogs to treatment concentrations, allowed them to mate and lay eggs. And then in the second phase, we maintained their offspring in the same mesocosms under the same treatment concentrations until they reached metamorphosis. So the NAFCs that we used were derived from a sample of OSPW taken from an active tailings pond. And we, of course, had a control and also picked concentrations of 5 and 10 milligrams per liter based on the findings of a preliminary toxicity test, as well as these concentrations being environmentally relevant. So they're within the range of NAFCs detected in wetlands within the oil sands region. And we set up 3 mesocosms per treatment. So I mentioned briefly that frogs were wild caught, and I wanted to kind of discuss more about what this involved. So wood frogs hibernate over the winter, and as they wake up in the spring, typically on the first warm, rainy night, before they do anything, before they'll even mate, or before they'll eat, they head to ponds to mate. So because I wanted frogs to mate in my mesocosm so that I could observe their mating behavior, it was essential that we were able to catch them during their spring migration when they were on their way to the ponds. So as you can imagine, there was a lot of pressure associated with this. If I didn't have frogs, I didn't have a project, I didn't have a master's. So my lab mate Lydia and I headed out to the woods one night, and we were able to collect all of the frogs. All it really entailed was wearing our headlamps and walking up and down roads surrounding wetlands in the QUBS area and picking up half-frozen wood frogs. But this was a very exciting night for a number of reasons. The migration of amphibians in the spring is massive. We saw so many salamanders, so many wood frogs, as well as other frogs. And the spring peeper choruses were so loud that we almost were yelling to communicate to each other. And I actually have a little video here of a peeper peeping. Oh, I might have to play it here. Oh, it might not play. Should I have a couple of videos? So. Okay. Is this the sound? No, it won't play. That's okay. I'm gonna try one more. No, okay, it's fine. I'll describe videos. The peepers on this log. Peeping, making a really loud, high-pitched noise. If you've ever been outside in the woods in the spring, you know what this sounds like. And I did look it up afterwards because I was curious, and spring peeper choruses can actually reach sound levels of close to 100 decibels, which is close to a concert. And I also found out later that those that study them wear hearing protection for that reason. I woke up the next day feeling like my ears were still ringing. And another kind of notable thing we saw, this down here. So wood frogs are known for being exceptionally eager to mate in the spring, to the point where males will attempt to implex anything that is moving. And this is actually a wood frog, a male wood frog implexing on what I think is a male green frog. So I'm not sure how that ended. And I was so kind of mesmerized by this event that I actually took my parents out this past spring to the pond behind their house to look for wood frogs and peepers. And this is a huge wood frog breeding aggregation that we stumbled upon. So each little dot here is actually a set of frog eyes and a separate frog. So there's like hundreds of individuals in this little area of wetland. And this right here is the eggs. So wood frog females will actually lay their eggs all together in one spot. And that is thought to function in keeping offspring, the embryos, warm as they're mating in the very early spring, and water temperatures can be close to 0. So I guess this is my PSA. If you ever kind of find yourself able to go out into the forest on a rainy spring night to look at or appreciate the migration of your local amphibians, I highly recommend you do so. So once we collected frogs, we were able to start the experiment. First, we exposed male frogs in pairs within mesocosms to treatment concentrations, and we exposed females individually in buckets. And this separate exposure lasted for 24 hours. And then we added females to mesocosms already containing males and recorded their mating behavior with security cameras up here. We built like these wooden kind of frames to stand above the mesocosms so we could record them kind of with an aerial view. And within each mesocosm, there were 2 males and 1 female, which reflects a sex ratio meant to mimic these natural breeding events where males typically outnumber females. And I was lucky enough to be able to go through 400 hours of frogs mating. And from these videos, we determined the probability and latency of amplexus, as well as oviposition, egg laying, and amplexus duration, and the number of amplexus events. Had more videos that were examples of what this looked like, but it's fine, I will describe again. So first of all, it took a lot of problem solving to figure out how best to record these frogs, because it's not something that's typically done. And we ended up going with security systems based on the advice of Chris Elvidge. It turns out the same properties of these cameras that allow them to record license plates and facial features at night are also very helpful for recording frogs at night. And I think a lot of field work, those of you doing field projects now know that a lot of it is being creative and using products in ways that manufacturers never thought they would be used. So this video was just an example of amplexus. The male frog goes over and amplexes the female. And this here are 2 frogs that are in amplexus. They eventually swim away and reveal a clutch of eggs. But this ended up kind of working with great success. We were able to pinpoint these behaviors down to almost a second. Oh, it's gonna play. So frogs entering amplexus there. The 3rd frog is over here, left out. And then these frogs will swim away and reveal this little clutch of eggs here, which will eventually expand. They are surrounded by a jelly coat that fills with water over time. So after the mating behavior phase, we removed adults and then assessed the clutch for fertility endpoints. So we removed the entire clutch and divided it into smaller subsamples of eggs. And then we placed it back into mesocosms within these colanders. This is an example of using a product again. And I also bought every last colander that Walmart had. And then we photographed clutches from above, and from the photographs determined clutch size, so the total number of eggs, as well as fertilization success, or the total number of embryos, eggs that were fertilized. So this was easy based on the fact that unfertilized eggs quickly turn white and never really progress past that perfectly circular stage. So we found no support or no clear support for our first hypothesis that NAFCs affect reproductive behavior or fertility. However, we did find some interesting trends. So this 4-panel plot here shows amplexus latency in minutes, oviposition latency in hours, the total time spent in amplexus in hours, as well as fertilization success as the proportion of clutch fertilized. And while we found no significant results, we did see that those exposed to the highest treatment concentration, so 10 milligrams per liter of NAFCs, showed lower amplexus latency, so mated more quickly, higher oviposition latency, took longer to lay eggs, more time spent in amplexus, as well as lower fertilization success. And chemicals that are capable of exerting estrogenic activity, interestingly, show similar trends, such as increased mating behavior, delayed oviposition, and reduced fertilization success, both through egg and sperm effects. So while not clear, our results could indicate that NAFCs are exerting some estrogenic activity and should motivate further investigation of these effects. So after the adult phase of the experiment, we maintained offspring in these mesocosms and sampled them weekly from April to June, and then daily in July once they began to progress to metamorphosis. And during each sampling day, we caught all of the tadpoles within mesocosms, which sounds a lot easier than it was. Sometimes it took hours looking for the last one. And then we photographed them on this sieve stand setup. And once individuals reached metamorphosis, which we characterized as Gosner stage 42, or the emergence of at least one forelimb, they were removed from the experiment. And from the data we collected on these sampling days, we determined survival, metamorphic success, the frequency of abnormalities, growth, and size at metamorphosis. And it's kind of overshadowed here in this arrow, but it's worth noting that some individuals took over 3 months from the time of hatching to reach metamorphosis. And I think the first day that I had one with arms was honestly one of the happiest days of my life. So it's worth noting, saying that I probably had a slightly different QUBS experience to a lot of you. I was lucky enough to be able to complete my research here last summer when there were a lot more COVID-19 restrictions in place. This did mean, though, that my field partner Sam and I were the only students on campus for a good chunk of the field season. We were living apart, cooking on our hot plates, and weren't really leaving campus at all aside from the odd grocery run. This meant a lot of solo swims in Lake Opinicon, which I will always be grateful for. And despite all of the differences, I really wouldn't trade the experience for anything. Those of you doing fieldwork now know how nice it is to be able to spend so much time outdoors, as well as kind of return to the same location outside every day. I got to witness so many interesting things and help a lot of turtles across the road. And one day I even saw a garter snake eating an American toad, which I'm very proud about, that photo. Once the frogs finally reached metamorphosis, we were able to conclude that we had significant support for our second hypothesis that NAFCs reduced offspring viability. So this first plot here, it's kind of cut off, but it shows survival, the proportion of clutch surviving over time. And we found that those exposed to 10 milligrams per liter NAFC were significantly over 10 times more likely to experience mortality during the experiment and showed a 42% decline in average survival relative to the control group. If we look at the second plot here showing the proportion of clutch at metamorphosis over time, those that did survive were significantly less likely to reach metamorphosis, showing a 22% decline in metamorphic success relative to the control group. And metamorphosis in frogs is highly dependent on thyroid hormone levels. As I mentioned previously, declines have previously been linked to differences in thyroid hormone ratios after exposure to OSPW, which suggests that these results may also be indicative of some thyroid disrupting effects. It's also worth noting that even though we did observe already some large declines in survival, these effects could be even more pronounced in the wild because wood frogs rely on temporary breeding ponds. And so delays in metamorphosis could lead to even larger mortality if individuals are not able to emerge from a pond before it dries up. It's so hot in here. I feel like I'm doing hot yoga. It's like hot presenting. We also found some growth effects. This is showing total length over time, again with the highest treatment here in orange. And we found that tadpoles raised in this 10 milligram per liter exposure group grew significantly more slowly. And similar declines in growth have been observed in leopard frog tadpoles exposed to these commercial naphthenic acids. And these were also linked to decreased glycogen and increased triglyceride stores, suggesting that NAFCs may be working, or naphthenic acids in that case, but possibly NAFCs, working through liver toxicity and effects on metabolism. So it's possible that the delays in metamorphosis and declines in growth that we observed are also caused by some liver toxicity. And if these effects were to carry over into adulthood, it could affect overwintering survival of offspring, as obviously metabolism and energy stores are very important during hibernation. So we also looked at abnormalities. This plot shows the frequency of abnormalities, again with NAFC concentration increasing from left to right on the x-axis. And we found that abnormalities were more frequent at every life stage, though they were only significant in the 10 milligram per liter control group at hatching. We also observed that multiple and severe abnormalities were more common in those exposed to 10 milligrams per liter of NAFCs. And some of these abnormalities included yolk sac edema, seen here, cranial, abdominal edema, and axal abnormalities in hatchlings, axal abnormalities in the form of tail kinks, as well as abdominal edema here in the larval phase, and limb abnormalities at metamorphosis. So I wanted to discuss these limb abnormalities a little bit more as they're observed here for the first time after exposure to NAFCs. This included what we've been calling micromelia, which is seen in the video and in the top image here. And that was individuals that developed with abnormally small limbs compared to body size. And then we also observed ectrodactyly, so individuals growing limbs that were missing digits. So frogs should have 5 limbs, or 5 digits, not 5 limbs, 5 digits on their hind limb, and we observed individuals with only 4. So similar effects have previously been observed in Chinese toads exposed to the thyroid disrupting element cadmium. And these are skeletal images from that study. As you can see, they observed individuals with missing digits, as well as this up here is a control. This is the cadmium exposed individual. You can see that they also observed this smaller reduced limb size effect. So it's possible that NAFCs are also causing these abnormalities through thyroid hormone disruption. And even though these individuals reached metamorphosis, if they're carrying these abnormalities into adulthood, it could obviously affect their fitness through things like decreased foraging success, reduced swimming speed, and maybe changes in predator escape ability. So in summary, our study represented the longest experimental exposure and first bi-generational exposure of an amphibian species to NAFCs. And it was also the first time that an amphibian was exposed to these OSPW-derived NAFCs under environmentally realistic field conditions. So we found no clear support for our first hypothesis that NAFCs disrupt reproduction or reduce fertility. But we did find strong evidence for our second hypothesis, suggesting that NAFCs disrupt reproduction and reduce mating success through reduced offspring viability. And it's important to note that recruitment of offspring to the breeding adult, every time I do that, to the breeding adult population is important to population health. And our findings suggest that fewer offspring will emerge from NAFC polluted ponds, and those that do may suffer reduced fitness as a result of these abnormalities and possible growth effects. Overall, our findings suggest that exposure to concentrations as low as 10 milligrams per liter during reproduction and development could have detrimental effects on native amphibian populations in the oil sands region. So science, of course, is a never-ending cycle, and there are currently there's currently more work being done to kind of address some of the questions that came up during the study. We took samples from adults, hatchlings, and metamorphs, and those samples will be analyzed for thyroid hormone levels in order to kind of address that question as to whether some of these effects are being caused by endocrine disruption. We also swabbed individuals at metamorphosis, seen here, and those will be sent to our collaborators at Waterloo and Barb Katzenbach's lab, who are interested in the role of the microbiome in the frog immune system and how NAFCs may be exerting some immunotoxic effects. And this is included just to show how tiny wood frogs are at metamorphosis, like the size of a fingernail. And as you can imagine, those were very easy to dissect. The spleen was microscopic. So in summary, there are currently 500 billion liters of OSPW being held in these reservoirs or tailings ponds. And this wastewater threatens the environment both through planned and unplanned releases. Our findings of developmental effects in frogs at concentrations as low as 10 milligrams per liter can contribute to the development of these environmental quality guidelines for NAFCs. And these guidelines will be essential to informing safe release criteria and environmental monitoring plans that will ultimately help protect the wildlife and environment of Alberta from oil sands pollution. Thank you to my lab, my supervisor, all of the volunteers who helped in the field, as well as with like data collection, measuring tadpoles and image J. I highly appreciate it. And thank you for listening.